All right, ladies and gentlemen, we're going to talk about now the final step in protein synthesis. We talked about transcription, now we're going to talk about translation. It's a little more in-depth than transcription, and there are some more foreign concepts. Transcription, where we made mRNA, was a lot more similar to DNA replication than translation is going to be. So, the synthesis of proteins is actually called translation. This is where our protein is actually being built. Now, the cell has to literally translate the base sequence of an mRNA molecule, which was made in transcription, into an amino acid sequence, which we know amino acids are the monomers of proteins. Now, where does translation occur? The site of translation, or protein synthesis, occurs in the ribosome or on the ribosome. The ribosome is going to facilitate an orderly linking of amino acids to create this protein. Now, during translation, the cell uses the information contained. Remember, the information contained within mRNA is contained within the order of the nucleotides, the A's, the U's, the C's, and the G's. Remember, T is only found in DNA. U's replace T's in mRNA. Okay, so let's look at protein synthesis as a whole, including transcription and translation, as we kind of learn more about translation here. So, the steps in protein synthesis. First and foremost, in the nucleus, DNA is transcribed into RNA via the process of transcription. The RNA is sent to the cytoplasm in the form of mRNA. Remember, that's the whole reason mRNA is made, is because DNA is meant to stay safely locked within the nucleus so that it runs the least risk of getting damaged. The mRNA finds a ribosome and attaches to it. Now, as each codon, remember, a codon is a series of 3 nucleotides, and it's actually how mRNA and DNA are read in the transcription process, in the DNA replication process. They are read in codons. Or I'm sorry, just the transcription process, not the replication process. So, as each codon of the mRNA molecule moves through the ribosome, which we kind of see happening here in this image, this big gray ball and this small gray ball are the ribosomes, and this yellow band is the mRNA running through it. The proper amino acid is brought into the ribosome by another type of RNA called tRNA. We're going to talk about tRNA just a little bit. The amino acids are lined up in the right order on the ribosome according to the codons and the order that they're read. So the ribosome hitches the amino acids together with peptide bonds, because the bonds that exist between amino acids are called peptide bonds, and these make proteins. Now let's talk about this transfer RNA, this very special type of RNA that brings in the amino acids after the ribosome reads the codon on mRNA. So the function of tRNA is to transfer amino acids. That's why it's called transfer RNA, right? There are amino acids floating around in the cytoplasm of the cell, and this tRNA, according to whatever codon is read by the ribosome, will grab that amino acid and bring it to the ribosome. The cell always has all 20 amino acids floating around somewhere in its cytoplasm, and millions of copies of these 20 as well. So the transfer RNA molecules are not all the same. Each type of tRNA molecule links to a particular mRNA codon with a particular amino acid. So the mRNA codon, all right, an mRNA codon on your codon chart that we used before break, codons translate directly to amino acids. That's why the outermost ring on your circular codon chart just has words. Those are the amino acids. And you're using the codon to figure out which amino acid that codon tells tRNA to get. So, as tRNA arrives back at the ribosome, it carries a specific amino acid at one end, and it's this end up here. Okay, this is where that specific amino acid is attached and being carried. At the other end, we have a new site called, I'm sorry, we're not going to name it just yet. At the other end is the nucleotide triplet, which we call an anticodon. So DNA and mRNA have codons. tRNA has anticodons. This AAG right here at the bottom of this loop, at the bottommost loop, that's the anticodon. This is going to complement the codon found in the mRNA. All right, so the mRNA's complement to this would be UUC. And then this tRNA would go and get whatever amino acid it's supposed to and bring that amino acid to the ribosome based on the anticodon of the tRNA and the codon of the mRNA complementing each other. So this is where the amino acid is attached, and here is our nucleotide triplet known as an anticodon. Okay, let's do an example, right? Because it's been a lot of talk so far. If an mRNA codon is UUC, so this is the codon found on mRNA, the molecule that was made by transcribing DNA and then found the ribosome in the cytoplasm. So what would this translate to as the amino acid? Well, if you use your codon chart, and I would urge you to pause this here and do that now, find UUC and then tell me what amino acid UUC codes for. And your answer should be phenylalanine. Now the tRNA that delivers that amino acid, phenylalanine, has as its anticodon AAG, which we see here and we saw in the last slide. And at the other end, phenylalanine is carried to the ribosome to build the protein. Now, each tRNA is used repeatedly to locate a particular amino acid and deposit it at the ribosome. It leaves the ribosome to go find another. These tRNAs are not used up in this process. They are just used over and over and over again. Now, it's really important that these codons, right, this series of 3 that our cells read mRNA in, is read correctly. Because just reading it off by one nucleotide can mess everything up. And I'll give you an example right here, maybe a more relatable example. How about this sentence? The red dog ate the cat. These are all 3-letter words, and it makes plenty of sense, right? I get it. Now, if the reading of the code starts in the wrong place, for instance, if we omit this T, if we forget that this T is here, her ed aga tet hek at. That is this sentence with that T omitted and keeping all 3-letter words as much as we can until the end where there are no longer 3 letters. This is the same thing that can happen in translation when we are reading codon by codon. This would not be the proper protein. If you think of each one of these as an amino acid, this is a functional good protein, and just omitting one nucleotide or missing one nucleotide in the codon, this is not a functional protein, and this could cause a lot of problems. Now, the result, obviously, is gibberish. The protein will be made putting the wrong amino acids in order, and it is unlikely that this protein will be able to function, or it will have some kind of detrimental function to the cell. Okay, folks, we're going to put it all together in the next one.